Okay, this is Carlos ARBIZU speaking. I'm a PhD student at the University of Wisconsin-Madison. Currently, I am working under the supervision of Dr. Spooner and Dr. Simon. And today I am going to talk about integrated molecular and morphological studies of Daucus. So this presentation consists of 4 main topics. First, background. Second, the molecular analysis. Third, the morphological analysis. And finally, a summary of both studies. So first of all, I would like to mention a brief background. We all know that carrots are a very popular vegetable worldwide. It's considered among the 10 most consumed vegetables in the world. It's also considered a nutraceutical food because it provides many health benefits to humans, to your heart, to your liver. It helps to prevent cancer, and it also has anti-aging properties. Cultivated carrot is grown on an estimated 1.2 million hectares annually worldwide. There are many hybrid seed companies, just like we can see here in the picture, that are looking for more resistances against pests and diseases, and also to increase yield, and of course other quality traits that are important and that can be found in wild carrots. Apiaceae, the family of carrots, is a taxonomically complicated family with many unnatural generic boundaries. There were many studies conducted before by Downey's group at the University of Illinois in the USA, where they found many species different from Daucus considered as in-groups within Daucus. So we conducted a morphological molecular analysis using 94 conserved nuclear orthologs. That means that we used 94 cos markers. We used 107 accessions containing 26 Daucus species and 10 close generic outgroups. We used the RBM method, the reciprocal best match, that is described by Wu and collaborators, using the ESTs of lettuce, sunflower, carrot, and the whole genome sequence of Arabidopsis and carrot. We obtained our DNA sequences using the 454 Roche platform, and it was possible to obtain DNA sequences ranging from 500 to 700 base pairs with an average of about 600 base pairs. We decided to conduct different dataset comparisons in order to select the best method how to handle this big data. First of all, we compared a dataset where we shortened the homopolymers to a maximum of 6 base pairs versus another dataset where we didn't shorten the homopolymers. Then we selected one allele per accession with the highest coverage, and we compared that dataset versus another dataset where we combined both alleles of Daucus using ambiguity codes. Then we compared the exonic content of our dataset versus the intronic content of our dataset. And finally, we decided to choose a reduced number of markers that best approaches the dominant topology. The dominant topology means that it refers to that topology that is obtained when we used all 94 markers. So we decided to use a reduced number of markers so that future workers can reconstruct the dominant topology in the near future. Of course, using less resources. So here our results are using maximum parsimony analysis. We can see here on the left the results when we used our data set where we shortened our homopolymers to a maximum of 6 base pairs. And on the right, our data set where we didn't shorten our homopolymers. So you can see that it was possible to obtain 2 main clades, similar to the work that was conducted by Downey and collaborators in the past. We obtained group A and B for both cases, and within group A, we obtained clade A prime, containing all Daucus species, all Daucus subspecies that have 18 chromosomes only. And the rest are the Daucus species that have 20, 22, and 44 chromosomes. You can see that there are not major differences between these two data sets. The bootstrap values, as you can see here, are very high, 100 in most cases. And interestingly, we found that Margotia gamifera was also an in-group here. When we compare the one allele dataset versus the dataset when we used the ambiguity codes, we found some differences. For example, in this case, when we used the one allele with the highest coverage, we found a clade that contains all the subspecies of Daucus that were collected in Libya and Tunisia. However, the bootstrap support is low in this case, and also we obtained a clade with very high bootstrap support, 100, containing all the subspecies of Daucus carota that were collected in Portugal and Spain. On the other hand, when we use the two alleles combined into one, using the ambiguity codes, we didn't find those clades. As you can see here, they are not present. However, the rest is the same. It's pretty much the same. The bootstrap values are very similar. And also, I would like to mention that here in clade B, we identified Daucus literalis, Daucus involucratus, Daucus pusillus too, but there is a conflict here between the Daucus guttatus and Daucus bruteri because they were not clearly identified. They were intermixed, so that's why we decided to name them as Daucus guttatus 1, Daucus guttatus 2, and Daucus guttatus 3 because we found 3 well-supported clades here. When we compare the exonic versus the intronic content, there were not major differences again. We obtained very high bootstrap supports, and again, the clade A and clade B, and also within clade A prime, so no major differences again here. This is very interesting because we could obtain with one marker the same topology when we use the 94 markers. So we identify the good markers that can give us the dominant topology, and this can save resources for other workers. However, I would like to comment here that there were 4 species that are misplaced here. But when we combine the 10 best markers, as you can see here on the right, we obtained the same topology with very high bootstrap supports again. So we can use this subset of 10 markers to reconstruct the dominant topology. We also conducted a Bayesian concordance analysis in order to analyze the gene-to-gene conflict in our data set. We used 94 markers, so each marker can give you a different topology. Each marker has its own history. So interestingly, we found that clade A is supported by about 35% of the genes, and clade B is supported it's supported by about 40% of genes, which is a normal value of support. However, here Daucus sahariensis and Daucus certicus are supported by about 70% of the genes, which is a very high value. So this means that there are minor discordant histories between these 2 genomes. On the contrary, between the subspecies of Daucus carota, the values are lower. So there are a lot of conflict between these clades, between these genes that form the clade. So I would like to mention that this molecular studies has already been published, and my professor David Spooner has copies of this, so if in case you're interested, you can take also a look to that paper. And now I would like to mention the morphological analysis that we conducted using the same species, using the same accessions that were used in our molecular study. So this morphological analysis was conducted to place the phylogenomic results in a practical taxonomic context. This study was conducted at the North Central Regional Plant Introduction Station in Ames, Iowa, the USA. We used 80 accessions, the same accessions that were used in our molecular study. It contained 14 Daucus species and 9 closed generic outgroups. We evaluated 40 characters from at least 3 individuals per accession. So here our results are. You can see that it was possible to identify, for example, Daucus muricatus using morphological studies. Also, Daucus littoralis, Daucus certicus, Daucus involucratus, Daucus sahariensis, Daucus glochidatus, Daucus pusillus, Daucus crinitus, and also the Daucus guttatus 3, Daucus guttatus 2. However, it was very hard to identify the subspecies of Daucus carota, the gummifer type and the carota type. You can see here letter E and F. It's not clearly identified. Also, Daucus guttatus 1 and Daucus capillifolius, the same. It was not possible to identify it using the morphological characters. I would like to mention now this study conducted by Euriso and collaborators. This is the maximum likelihood tree of Daucus wild and cultivated species. Interestingly, they could differentiate between the wild and cultivated carrots. Not only that, but they also differentiated the localities where those wild carrots were collected: Central Asia, Greece, Middle East, North Africa, Europe, North America. Also, they differentiated between the Western and Eastern cultivated carrots. In order to do this work, they used about 4,000 SNPs. So I would like to mention here how powerful the SNP markers can be to differentiate the Daucus carota. So that's why we have decided, our group has decided to continue our studies using SNPs and GBS data. Now I would like to mention this work conducted by Spooner and collaborators, where they identified the 2 types of Daucus carota, the gummifer type and the carota type. So in summary, our molecular study is the most complete study of Daucus to date relative to the number of accessions and markers, showing excellent topological support. Margotia gummiferase and in-group in Daucus, which is a new finding. The best data set was to use one allele with the highest coverage. There were no differences when we compared the other data sets. There were misidentified accessions in the Daucus US germplasm bank. We selected the best 10 cause markers out of the 94 to reconstruct the dominant topology. Daucus capillifolius and the subspecies of Daucus carota are poorly separated morphologically. The subspecies of Daucus carota need more study, and we are using GBS data for this. We propose an additional replicated morphological study in an appropriate environment for the subspecies gummifer. The status of Daucus broteri and Daucus guttatus is unresolved so far because our present data supports 3 species. SNPs are a very powerful markers to distinguish the structure of Daucus carota. Finally, I would like to thank you very much, Dr. David Baum and Dr. Cecilia Neff for their advice in this work, the USDA for its financial support, and the University of Wisconsin-Madison for giving me the opportunity to be here at this wonderful meeting. And I'm happy to take questions now.
When you build the trees and you pick an allele instead of using the heterozygotes, how did you pick alleles? And did you run your trees picking different alleles and running the topology, or what did you do?
Yeah, so we picked the alleles using the highest coverage that we could obtain. So we obtained 2 alleles for most cases, and for one data set we selected 1 allele with the highest coverage. And for another data set we combined those 2 alleles into 1 using the ambiguity codes for the DNA.
Yes, Thomas. Yes.
The classical taxonomy discriminate between species on the basis of crossability. What you means to Capillifolius? I know that Capillifolius is crossable with all Daucus carota subspecies, and your results say Capillifolius is in the same clade, and so I think
using molecular markers and morphological descriptors, it was not possible to clearly identify Daucus capillifolius. So that's why in our recent paper using morphological characters, we have decided to rename Daucus capillifolius as a subspecies of Daucus carota. Mm-hmm.
Just a quick question related to that. I'm surprised that you say that it's difficult to differentiate Daucus capillifolius, I'm losing my word, capillifolius and Daucus carota subspecies, because they look quite different.
Yes.
I mean, and the related question is also the number of morphological traits that we use for this kind of study, and your canonical study, maybe some traits are heavier role in the analysis than others. And my question is, among the 40 traits, maybe there are some that are more essential than others to look at the organization.
Right. Well, Daucus capillifolius clearly has a different color flower. It can be easily differentiated, but when we use all the 40 characters that we evaluated, I mean, in this canonical analysis, it was not possible to differentiate them. And with the molecular markers, we couldn't differentiate them either.
And can we think that we could have a short list of morphological traits that are really the, common, important ones? The top 10? No?
Sure. But, I mean, when we try to put in a practical content the morphological analysis and the morphological analysis, we didn't find a clear pattern to identify the Daucus capillifolius.
Okay, thank you.